Good morning, everyone. Thank you to the organizers for giving me the opportunity to speak today. We're going to shift gears a little bit and get into the cultivation side of things. And for those of you that perhaps don't have any experience in seeing how cannabis is grown, I think this will be a great chance for you to learn the procedures. But more importantly, I'd like to talk about a major threat to the cannabis industry, and that is a disease that's called Hop Latent Viroid. And you'll see some of the features and characteristics of this particular disease today. So eat your heart out. This is my work environment. This is where I spend 2 days a week in the greenhouse looking at cannabis cultivation. This is a greenhouse operation in British Columbia, Canada. And all of the data that I'll present today comes from an environment such as this. And you can see highly uniform, sophisticated and hydroponically cultivated cannabis production, which of course is the high THC or marijuana. This is what the pathogen or disease, Hop Latent Viroid, can do to plants. On your left is the stunted, infected plant that has Hop Latent Viroid, and on your right is the healthy plant. And you can see right away that you've got a significant reduction in growth, which you'll see later correlates to a reduction in the amount of cannabis flower as well as THC that comes out from these plants. Here again is a really nice comparison. Infected on your left, healthy plant on your right. Smaller leaves, darker leaves, not as vigorous growing as what you would expect in a plant that does not have this particular pathogen. What exactly is Hop Latent Viroid? It's a RNA molecule that exists in these particular plants. They happen to be the absolutely smallest pathogenic organism that we know of, which is 50 nanometers, which is 5/100th of a micron. It is so small that we can't even see it other than with an electron microscope. It's a pathogen that does occur on other plant species such as potato and chrysanthemum, peach, and so on. It does not affect humans or animals. It seems to be restricted primarily to plants. And at this point in time, I consider this to be the COVID of the cannabis world. And you'll see why in a few minutes. So it was first reported in California back in 2019. And plants there were stunted, not well developed. The flowers were not doing well. The plants were not rooting well. And the symptoms does vary depending on what stage of the plant we're looking at. And I'll describe some of this as we go through the presentation today. So here you see a plant that's in full flower. On the left-hand side is the stunted plant that is obviously affected by this Hop Latent Viroid, produces much smaller flowers, and overall yield has gone down. This is what the pathogen will do when it infects a plant and the inflorescences or flowers are developing. You can see that the infected flowers are much smaller and will produce much less cannabinoids overall. When you actually look at them side by side, morphologically they're no different other than the fact that they're considerably smaller in size. In some genotypes or chemovars, we do see a symptom such as this. It almost looks like a chrysanthemum type flower. The yellowing that you see is not common. This is not something that you'd like to see in a cannabis flower. You're seeing a lower production of chlorophyll. And in addition, the roots are affected. You can see the healthy compared to the infected root systems. The plant isn't doing well, it's not growing well, and as a result, it's going to produce less flower and less yield. And that's a really good comparison of how the root systems have developed on these two plants. Growers know that when they take cuts to propagate cannabis, the cuts that come from infected plants do not root well. So here again, what you're seeing is the underside of a rooting block, the infected one on your left, the healthy one on the right, very reduced root production. So this is quite an interesting pathogen. It affects the flowers, it affects the growth, it affects the roots, and as you'll see later on, it affects the cannabinoids. So obviously when you have a plant that's producing a poor root system, it's not going to grow as well, it's going to be obviously stunted, and it's not going to be taking up as much nutrients as it should. And in addition, I'll show you at the end of the talk that these plants also seem to have increased susceptibility to other pathogens. So this is where the COVID analogy comes in. People that suffer from COVID usually have other disabilities or other issues that affect the liver or the heart or other parts of the body. When these plants have Hop Latent Viroid, they may also be reduced in their ability to fight off other diseases. Now here's a really interesting slide that compares one plant in which one side of the plant has the Hop Latent Viroid, which is the bottom side, and the top side is actually healthy. And so here we can make a direct side-by-side comparison. And you look at the numbers, you'll see where it says Hop Latent negative, the THC concentration is 27.8, and the terpene concentration is 1.3. You move down to the lower left where it says Hop Latent positive, your THC concentration has dropped to 15.9%, almost a 40% reduction, and your terpene levels have dropped to 1.1, about a 15% reduction. Now this is pretty significant for those of you that work in the industry for medicinal or pharmacological use. When you have a plant that's infected with Hop Latent Viroid, it's going to produce less cannabinoids, less THC, and less terpenes. Here's a very good comparison of what a harvested plant will produce with regard to absolute yield. And so once again, healthy on the left and Hop Latent infected on the right. You can see the smaller size overall. Way lower yield, way lower dry mass as a result of this particular pathogen. Now the reason that the cannabinoids are reduced is shown here in this schematic, where you have a nice healthy trichome on your left and a trichome that comes from a diseased plant on the right. And what we see, and I'll show you this in the next few slides, is a reduction in the size of the trichomes and in the volume of material that they can produce. And so because these trichomes are primarily synthesizing cannabinoids and terpenes, when you have a reduced shrunken trichome, clearly that's going to impact what's being harvested from that plant. So here's a comparison. This is using a scanning electron microscope where it says healthy, are the healthy bracts, comes from the flowers that are not infected by this pathogen. And over on the other side where it says infected, you can see overall that the little blobs, the trichomes, are smaller and they're stunted in terms of how they develop. And here's a close-up again where it says healthy. These are the trichome glands or the bulbs where all these cannabinoids that we heard about from Dr. Russo and other speakers are contained. And on the bottom is the inflated trichome gland. Look at the infected plants where the trichomes are shriveled, much smaller in size, and obviously are going to produce much less cannabinoids and terpenes. And so this is why this disease, this pathogen, is of such concern. When you see this impact, reduction in your cannabinoids, it's clearly a cause for concern. Now we've collected some data on a number of different strains or genotypes of cannabis, and what this table is basically showing you is how this particular pathogen impacts plant height, which is the first column, the flower stem length, the second column, flower fresh weight, the third column, and then finally THC. And you can see overall that the range of reduction is anywhere from 27 to 31% lower. THC is down 28.8% overall. Varies by strain. There's some strains that you see a 12% reduction and other strains almost a 40% reduction. So there definitely appears to be a genotype effect in terms of how these plants respond to this pathogen. Now, in order to detect this particular organism, this pathogen, we use what's called a PCR method, RT-PCR. Again, very similar to what was developed for analysis of COVID. And what you're seeing here is a gel, and the bands, the white bands that you see, the multiple bands, indicate the presence of the pathogen. So I'll be showing you a number of these slides that illustrate wherever you see the white bands, it tells you that the Hop Latent Viroid is present in that particular sample. So here what we've done is we've compared a range of different types of cannabis plants, different genotypes, and basically what this is telling you is that Hop Latent Viroid was present in all of them using these particular PCR primers. The arrow where it says root sample indicates the presence of the viroid in the roots of the plants as well. And this becomes significant, as you'll see in a few minutes. So how does this viroid spread? Again, very similar to COVID. It spreads through contact, through handling of plants, through any tools or instruments that growers are going to be using to take care of their plants. It can also move through root contact, and I'll indicate how that happens. It can move through water. If there's recycled water that's being used in the facility, Hop Latent Viroid can be found. We don't know if it's transmitted by insects yet, and we do know that it's transmitted through seeds. And that's a really important implication for the industry as well. So this thing does really get around extremely well through handling and through contact and through water and through seeds. So here's an experiment we did where we artificially introduced the Hop Latent Viroid using a Q-tip that was dipped in infected sap. And the lower panel shows you that after you've introduced the viroid, within 2 weeks, it can be detected, first of all, in the roots. It moves from the upper stem into the roots within a period of about 2 weeks. And then when you come back and revisit that plant at 4 weeks and 6 weeks, what you find eventually is that the entire plant is infected. So again, what you're seeing on the lower left, those bars indicate the presence of the viroid. And over on the right, it indicates how this particular organism is spreading. So from the point of inoculation, which was the top, it moves down into the roots, and then from there upwards into the rest of the plant. And that becomes really important from a management perspective. So this is what we know about Hop Latent Viroid, is that it moves systemically inside the plant through tissues that are called the phloem. The phloem is basically the plumbing system in the plant. And so when you've got something in the roots, the plumbing system will make sure that that viroid moves up into the leaves and, of course, into the flowers. So we also conducted another experiment where we placed an infected plant next to a healthy plant inside these cloners. So what we were trying to show here was the spread of the viroid through water. The two plants were not in contact with one another, and the nutrient solution was being circulated. And lo and behold, the little plant that you see on the left, which is marked infected, spread the viroid over to the plant on the right, simply through water transmission. And so we demonstrated that again using these two plants that were placed in a flat. The plant on the left was infected, the plant on the right was not. We flooded water through those two plants, and lo and behold, we had a 20% transmission rate from one plant to the next. So this is a common way that growers will water their plants, is to flood them. And the idea here now is that the viroid could move quite readily through the watering system. This was an interesting experiment where we took a bunch of clones, cuttings made from 3 infected mother plants. And as you can see, the roots are starting to emerge on the bottom of the cuts at varying levels. What was really interesting was that in each and every cut or clone that rooted, we found Hop Latent Viroid to be present. And that's shown on the gel on the bottom. Every sample had those white bands, which indicates that the viroid was present. So this is another very important way by which this pathogen moves, is through cuttings that are made from infected plants and are rooted. So here's just a summary. When you've got the viroid being introduced into the roots, it'll spread upward into the rest of the plant. I'll show you some data in just a minute that it gets into the flowers, it gets into the male flowers, it gets into the female flowers, and it gets into the seed. And again, that becomes real problematic as far as how this disease may be distributed throughout the cannabis industry. We observed that when plants go into flower, and those of you that may not be familiar, for cannabis to flower, the photo period has to be switched from 12 hours of light to 12 hours of dark. And the minute you start to do that, what we observed was that the concentration of the viroid started to increase. and it started to move upward into the flowers. So what you're seeing here is a plant that's on the left that is receiving full light, and on the right is the one that goes into flower. And when you start looking for the viroid, you see a dramatic increase, as you see with the presence of these white bands, in the plants that are under 12 and 12 photoperiod, which is the flowering period, compared to the panel on the right, which received full day length. So we think that the switching of the plants from a vegetative stage into a flowering stage is causing that particular viroid to move upward into the flowers. We don't know why just yet, but it's an interesting and has implications for seed transmission. In fact, when we analyzed dried hop flowers that are out there in the marketplace, we went to 12 or 15 different dispensaries. 40% of the dried flowers that we collected from dispensaries had Hop Latent Viroid present in them. Now, as I mentioned, this is not a human pathogen, so it's nothing that we need to be concerned about. What you need to be concerned about is the fact that the presence of the viroid in those flowers is going to alter the cannabinoids, the terpenes, and the ratios of those cannabinoids. And that obviously has implications for medicinal use. So here's an example of where we detected Hop Latent Viroid in male flowers. Male flowers are used primarily just for breeding, and you can see the panel on the right where we looked at floral tissues, anthers, and other parts of the flower. It was present. So this is data that we got from Toomey Genomics, where they made crosses between plants that were positive for Hop Latent Viroid and male plants that were negative for Hop Latent Viroid, and they did the opposite across as well, where they took female plants that were negative for Hop Latent and crossed them with male plants that were positive. And what they found when you do these crosses is that you can find the viroid in the seed, and on the seed coat as well. So here's the data that they shared with me. The orange bars indicate the presence of the viroid in seed, where the female plant was infected. And the bars on the right show you the presence of the viroid when the male plant was infected. So you can see obviously a higher transmission when the female plants are infected, but also transmission when the male plants are infected or as a source of pollen. And when you look at the actual numbers of seed infection, it can be as high as 43%. That's a pretty high transmission rate through seeds. It varies a lot from plant to plant, but that is indeed quite a significant observation. The good news is that there are differences amongst cannabis chemovars or genotypes in response to this disease. This shows you the relative frequency of infection. The bars indicate how much infection you had, and you can see with the lower bars, the two arrows on the bottom, they seem to be some genotypes that have a better ability to withstand this disease than others. Very good news for breeders. We conducted an eradication program. An eradication program is where every single plant that's infected is removed from a growing facility. And what this bar graph is showing you, this line graph, is the removal of plants from June of 2022 until January of 2023. And you see rises in Hop Latent Viroid infection, and you see declines in viroid infection. But the horizontal line, or the line that's pointing downwards, indicates that eradication can work if it's done successfully over a very long period of time. Every single plant that's infected needs to be removed from a growing facility. There is options for tissue culture as a way to eliminate this particular viroid. What I show here are two panels. The top panel is where you can use meristem culture. On the bottom panel is where you use nodal stem culture to produce plants. Meristem culture is the preferred way in order to try to obtain plants that are free from this disease. There are some things that are needed to be resolved before tissue culture can be used. The plants really need to be tested rigorously to make sure that they don't contain the viroid. But it is a possibility that meristem culture could be a way to eliminate this particular pathogen. How well does this viroid survive? Unfortunately, again, it is quite a bit like COVID. This is a slide that indicates It survives on gloves. In this case, it survives on gloves for up to 5 days when the gloves are left at room temperature. It survives in leaves. In this case, it survives in leaves for up to 4 weeks, leaves that are left in the greenhouse on the floor. It also survives in various types of root tissues, and I'll show you a couple of slides that indicate that this viroid is extremely stable. It is extremely stable. For example, when you look at temperature, this was where we took small root tissues that were infected by the viroid and heated them up to 70 degrees centigrade for 30 minutes. And the white bars, again, indicate the presence of the viroid. Even heating that tissue to 70 degrees for 30 minutes did not destroy the RNA. This is an example where we took UVC light, UVC irradiation, and we exposed leaves and roots for 5 minutes, long enough to cause a severe burn on your hand, and the viroid was still intact. You can see the bars are present. So it's UV stable. And more than that, it can tolerate things like bleach and xylitol and Virkon. These are chemicals that normally, as disinfectants, would eliminate COVID. They have absolutely no effect on this particular viroid because of the stability of the RNA. That's not to say that these chemicals cannot be used in a cleanup operation, but it indicates that neither of these chemicals actually destroys the RNA. Well, here's the good news. I have to end the talk with a little bit of hope because everything I've said so far indicates this thing is so stable. Nucleic acid enzymes, things that are called nucleases, in particular a particular type of nuclease that's called RNase 3, will actually destroy the RNA of this viroid. So here what you're seeing, the vertical arrows, the blue arrows, indicate where we added the enzyme, the nucleases, and it totally disrupted and destroyed the viroid. What does this mean from practical purposes? I have no idea. What we do know, though, is that there are specific enzymes that could be used somewhere on the line to actually destroy this particular this viroid. So quickly, I'll show you how Hop Latent Viroid infected plants are more susceptible to other diseases. This is a disease that's called Fusarium oxysporum wilt, and you'll notice where it says Hop Latent minus and Hop Latent plus. The one that has Hop Latent Viroid actually picks up more Fusarium infection than the plant that's not infected. And here you can see a very nice comparison of two flowering stems. The one on the left with the white mycelium is the plant that was infected with Hop Latent. The one on the right did not. And you can see how much disease they develop as a result of the infection by the viroid. Here's some data to illustrate. The blue arrows basically tell you that those are the lines in which Fusarium was increased as a result of the presence of the Hop Latent Viroid. And that's of concern because it indicates that those plants may be more susceptible to other pathogens. And lastly, we looked at powdery mildew, which is another important disease in the industry. And what we found again with powdery mildew is that when you have Hop Latent Viroid present, you get more infection with powdery mildew than you do when the plants are healthy. And the graph on the bottom indicates the green line is the infected plant and the blue line is the plant that does not have Hop Latent Viroid. And so this is my last but one slide, and I'll show you the really amazing contrast between a Hop Latent infected plant with powdery mildew and a Hop negative plant with some powdery mildew, but not a lot. So again, this is a predisposition to other diseases that may be present in the cannabis operation. So there still are a number of unanswered questions. We can probably discuss those over the break. I think we're almost out of time here, but what is the basis for differences among cannabis chemovars or genotypes to this disease? We're not really sure yet. Can we develop a screening method for this pathogen? Possibly. Do plants that grow in soil get less viroid? Maybe. Everything I showed you today was in hydroponics. There's a phenomenon called latent infection in these plants. We need to understand more about that. And certainly things like mycorrhizae, will they protect plants from Hop Latent? We don't know that. So I'll end here with an acknowledgement to the research assistants and students that help you in the research, as well as funding agencies and our collaborators. and various government agencies for providing funding. And thank you very much for your attention.
Thank you, Zamir. That was very enlightening. I've heard that some tissue culture labs are not successful in eradicating HLV. Are there tricks that you could indicate to us to increase the success rate?
Yeah, so that's a really good question, and I think it varies a lot from lab to lab. I think the key is to, first of all, only obtain tissues from the tissue culture plant that you're going to use that's at the very top of the meristem. The other thing that has to be done is the plants that you use should not have high levels of the viroid. In other words, you need to try to suppress the spread through whatever mechanisms might be available, whether it's through the use of antiviral chemicals or in some cases, some researchers have stated plants that are incubated under cold conditions will actually have less replication of this viroid. So you put the plants in cold, you take only very small meristematic tissues, and then you really need to test. You need to test, you need to test, you need to test, because this viroid does stay latent. Again, similar to COVID, it's in your body, but it doesn't manifest unless there's some stressful condition or some environmental condition. The plants need to be completely tested well to make sure that they're clean before we actually release them. So there's definitely a lot of effort going into that, and I think that's going to be one of the ways in which we can eradicate this disease. Yes. 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 Right, right. So the nuclease approach is not going to replace meristem culture or anything else that the industry wants to look at. So it's going to be something right now that's strictly hypothetical. The slide that I showed you here was actually taken about 5 days ago, where we wanted to run the experiment to prove the concept that there are enzymes that will destroy the specific RNAs in this viroid. How you apply it, where you get it from, how you use it is completely unknown. Are we going to genetically engineer a plant to produce nucleases? Are we going to insert nucleases into the plant? Are we going to use a bacterium to deliver the nucleases? We don't know that yet, but all we do know is that there is one approach that can hypothetically destroy this viroid. Now, it could be something that's used as a cleaning agent. You apply it to the surface like you would UV light and remove any viroid that's on the outside. How you actually introduce that into the plant is something that I think is going to still require a lot of conceptual design and experimentation.
Dr. Punja, a very informative talk, as always. As we know, there are some plants that are capable of keeping the level of HLVd molecule at bay, particularly in the root area. So it seems that there are some effective defense mechanisms present in these plants. And as you know, I'm interested in genetic resistance to diseases. Was wondering if you might have any opinion about the nature of these defense mechanisms.
Yeah, so the question was, as I mentioned earlier, there are plants or cultivars or chemovars that differ in their ability to withstand this viroid. Again, very similar to humans and COVID. Some people get more of it, some people get less of it, and we only are now beginning to understand that. In plants, it's very similar. Some plants can fight this thing off much better than others. There are definitely genetic mechanisms that determine that. We're not quite ready to get into that yet, but Kevin McKernan from Medicinal Genomics will speak after break, and he's going to talk about some strategies that can be used from a genetic perspective in the plant that will allow us to mitigate the spread of this particular viroid. So I'm going to leave it with Kevin, and because we haven't approached that yet, but there is hope. I don't want to leave this room with a doom and gloom, even though it is a very important disease. It is a severely important disease, but Kevin's going to talk more about the genetic basis for that.
Dr. Zamir, thank you very much for your research. Just so I understood you, with the Virkon S 2% solution, Is it still worthwhile to soak, say, pruning shears in the solution prior? Is that still a viable method for cleaning?
Absolutely. Any sanitation approach that you take, whether it's ethanol, bleach, or Virkon, you should still continue doing. I think what I was trying to mention here was that they don't destroy the RNA. They aren't a curative, but they're definitely worth protecting, just like gloves, right? Wash your hands, wear gloves. That protects you, protects the plant, doesn't eradicate it. So definitely, Virkon seems to be one of the best products out there. I would say bleach is also pretty good. Ethanol, maybe not so much. But definitely dip the tools on the hands, on the surfaces, anywhere where the viroid can come into contact with the plant surface.
Just a follow-up question. What percentage of bleach would you advise?
You know, I don't think you want to go more than 10% because it's just so caustic to everything around you, and that's the problem with bleach and the smell. 20% would work better, but I would never suggest you do that. It's too high.
Thank you.
I think there's one there, and then maybe we can do one before we go for break. I'm going to be around during break as well, so if we need to break, we can certainly do that.
Hi, Dr. Punja. My name is Bill Graham. I'm with Microclone Tissue Culture. We've talked online, and I wanted to ask, we promote a cultural program based on vigor, and that is taking tiny introduction cuts from the tips of the very best veg plants every cycle to create the next generation, and cleaning the tools and everything in between, with the intent sort of to be to capture those small pieces of tissue that would be viroid-free for the circumstance that they had just outgrown the viroid, and to prevent back contamination. And it seems to be reducing the presence in the entire population, even if it's just a few percent at a time, over and over. And your thoughts on that?
Yeah, so definitely a combined approach is going to be extremely important. So continuing to use the sanitation products, looking at the meristem tips for propagation. Now, some growers will try to use the uppermost part of the plant and use that as opposed to the bottom part. What we've seen from our data is that when the viroid gets down into the roots, the first place it goes is actually up to the top of the plant. And so taking those cuts for propagation, it may not be the best idea. But for meristem growth, and for those of you that are not familiar with meristems, the meristem is a very, very, very tiny piece at the top of the plant. And the idea is that as the plant grows, the viroid doesn't get all the way up into the top. And so by harvesting that meristem, you're one step ahead of the viroid. And that's the theory behind it. But you got to be careful because I think testing is going to be super important.
That'll be the next part. We'll be talking to you about the most strategic testing protocols.
So, I mean, definitely RT-PCR is going to be the method of choice, and that's what's out there right now. And there's many labs that actually perform the diagnostic testing for Hop Latent.
That's it. I meant the wet tissue. Oh, go ahead. I'll talk to you later.
Yeah, yeah, okay. Well, let's follow up later. Thank you. All right, thank you.